Hey everyone, and welcome. Today we are diving into maybe the biggest question there is. How did life even begin? How in the world did a lifeless, rocky planet give rise to, well, everything we see around us? We're going to explore how chemistry, one step at a time, solved the incredible puzzle of becoming biology.
Okay, so let's start with a really simple but kind of profound question. What is the actual difference between a rock and a rabbit? One is just inert matter, simple chemistry. The other is this unbelievably complex, information processing, self-replicating thing. That gap, it feels impossibly huge, right? But what if it wasn't one giant leap?
See, the secret is that the beginning of life wasn't some single magical poof. It was much more like an epic detective story. Nature was faced with a series of massive, distinct problems, and it had to solve each one to unlock the next piece of the puzzle. So today, we're going to follow the clues.
First up, where do you even start? Well, with the most fundamental problem of all, energy. You can't build anything without power. So on a chaotic, early Earth with no sunlight-eating plants or anything like that, where did the raw juice to kickstart the chemistry of life even come from?
Well, the answer might be hiding deep, deep under the ocean, at these incredible places called alkaline hydrothermal vents. Just picture this. You've got warm, alkaline water, full of hydrogen, bubbling up from the seafloor. And it's mixing with a cold, acidic ocean that's packed with carbon dioxide. This created a very, very special situation.
This difference in acidity, that's the whole key right there. The vents had these thin, porous mineral walls that acted like a natural membrane separating the two different fluids. This created something called a proton gradient, which is basically a natural battery. Think of a hydroelectric dam, right? It uses the difference in water levels to generate power. These vents did the exact same thing, but with a flow of protons, creating a steady stream of energy. And it was powerful enough to start forcing hydrogen and CO2 together to build the very first organic molecules.
Okay, so we've got energy, and we're making some simple organic molecules. That's great, but they're just floating around in a huge ocean. For them to really do anything interesting, you got to get them together in one place. You have to solve the container problem.
And this is where chemistry and, basic physics just does the work for you. Turns out certain molecules, like fatty acids, the stuff that makes up soap, they have this love-hate relationship with water. So when you just dump them in, they automatically arrange themselves into these tiny little bubbles, or vesicles, to hide their water-hating parts. It just happens. No instructions needed. Nature has all these simple ways of building walls and creating these little protocells all on its own.
And once you have a container, it's a total game changer. I love this quote from the biologist Michael Levin. The cell is a machine for exploring what is chemically or evolutionarily possible. By creating a boundary, you've essentially built a tiny little laboratory, a protected space where chemistry can start to run wild and experiment.
But a container with some random molecules inside, that's still not life. And that brings us to, honestly, the biggest hurdle of them all, the information problem. If you want to have evolution, you absolutely need a system that can store instructions, copy them, and pass them on. You need a blueprint. And just look at the difference here. On one hand, you've got the prebiotic world. It was just a chemical mess, right? A chaotic soup with thousands of different molecules, all bonding in sloppy, unstable ways. And then you have modern life, which is super clean, super ordered, built on just a few core molecules, with very specific, stable bonds. How did you get from that chaos to this kind of order?
Well, a really elegant and powerful idea is called the RNA World hypothesis. See, in our cells today, DNA stores the information and proteins do most of the actual work. But RNA is kind of like a Swiss Army knife. It can store information, like DNA, but it can also fold up and act like an enzyme to make chemical reactions happen, just like a protein. The idea is that early life used RNA for both jobs, which dramatically simplifies the puzzle of how you get the first self-replicating system off the ground.
Okay, so let's recap. We've got energy, we've got containers, and we've even got a plausible way to store information. Life should be off to the races, right? Well, not exactly. For the next 2 billion years, life on Earth was basically just simple bacteria. It just stayed there. Why? Because it hit a brick wall, a massive energy barrier that stopped it from getting any bigger or more complex.
For a long time, scientists called this period the boring billion. But it turns out it was anything but boring. It was more like a slingshot period. The oceans had really low levels of nutrients, which created this intense pressure cooker environment. And that stress forced simple cells to innovate, to develop all the key tools for complex life, things like a nucleus and even sexual reproduction, getting everything ready for the main event.
And then, the event that finally shattered that barrier, it was just revolutionary. It's something that might have only happened once in the entire history of the universe. One cell swallowed another one. But instead of digesting it, it put it to work. That captured cell became the mitochondrion, a dedicated power plant living inside another cell. And the result was an insane energy explosion. Suddenly, a complex cell had tens of thousands of times more energy for every gene than a bacterium did. This just obliterated the scaling barrier and finally made big, complex multicellular life like us possible.
So, when you look back at this incredible journey, from simple chemistry to complex biology, it kind of makes you step back and ask a bigger question, doesn't it? If life is a process, not a thing, then what is it? How do we even define it? And how would we recognize it if we found it somewhere else?
This is where this really cool, kind of mind-bending idea called assembly theory comes into play. It's a way to actually put a number on how complex something is. Think about building the word abracadabra. The shortest way isn't to type all 11 letters. It's to make abra, copy it, and then add cad. That's a shorter assembly path. The theory measures an object's complexity by finding that shortest possible recipe. The goal is to give us a universal test to tell the difference between something made by life and something made by random chance.
And here's where it gets really wild. The theory leads to this incredible prediction. There seems to be a complexity threshold, right around the number 15. Any object with an assembly number higher than 15 is so ridiculously unlikely to form by chance that it pretty much had to have been created by a process that involves memory and selection. In other words, by life. This number could literally be the signature that life leaves on the universe.
So you put it all together. Life isn't a miracle spark. It's a series of puzzles that chemistry predictably solves. And now we might even have a tool to find its unique fingerprint out there in the cosmos. All of which leads to one final, absolutely thrilling question. If life is just what matter does when it evolves, where else has it happened? Thanks so much for joining me.